In this video, we'll discuss what CRISPR-Cas9 actually is and where it comes from, how bacteria use it as an immune system, how we repurpose it to edit virtually any gene we choose, and at the end, we'll discuss some limitations of CRISPR-Cas9, as well as some common concerns. Let's start with what CRISPR-Cas9 actually is. CRISPR-Cas9 is an ancient immune system found in bacteria. Bacteria can be infected by viruses called bacteriophages, which, by the way, could not look more diabolical. And CRISPR-Cas9 is how the bacteria defend themselves against these infections. Now, CRISPR-Cas9 has two major components, CRISPR and Cas9. The CRISPR component is a specialized region of the bacteria's DNA, and the name CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats, which, yes, is a nightmare of a mouthful. But it actually tells you quite a bit about the CRISPR component. So let's quickly break down the name so we can understand what it's telling us. Let's start with the short palindromic repeats part of the name. Now, short and repeats are pretty self-explanatory, but the word palindromic may not be. But at some point, you may have learned about palindromes, which are words spelled the same forward as they are backward. Like the name Bob. You spell it in this direction, it's B-O-B. You spell it in the other direction, it's still B-O-B. But since we're talking about DNA, a palindrome in this case refers to the actual sequence of DNA, the nitrogenous bases, the A's, the T's, the G's, and the C's. So let's look at a quick example of a short palindromic repeat so you can see what I'm talking about. Now, if you look at the top sequence, it's GAATTC, which, as the arrow indicates, you read from left to right. Now, if you look at the complementary sequence that's on bottom, this sequence, you'll be reading from right to left. And as you can see, it's the same sequence, GAATTC. So since you can read one sequence in one direction and read the other sequence in the other direction, and they're both the same, that makes this a short palindrome. And because they repeat, they're short palindromic repeats. Now, the rest of the name is also pretty self-explanatory. It tells you that these short palindromic repeats are clustered together and regularly interspaced. So that's the CRISPR component, the DNA component. Let's now turn our attention to the Cas9 component. Now, Cas9 is a protein. Specifically, it's a type of enzyme called an endonuclease. A nuclease is a specific type of enzyme that cuts nucleic acids, such as DNA. You can think of nucleases as molecular scissors. Now, there are exonucleases and there are endonucleases. Exo means outside. So these scissors cut the ends of DNA. Endo means within. So endonucleases, like Cas9, will cut DNA somewhere in the middle. So how do CRISPR and Cas9 provide immunity for bacteria? Like I mentioned earlier, bacteria can be infected by bacteriophages. Now, assuming the bacteria survives this infection, different Cas proteins like Cas1 and Cas2, Cas9 does its job later. These other Cas proteins will use their endonuclease activity to take fragments of the viral DNA and incorporate it into the CRISPR region of the bacteria's DNA. Specifically, these fragments of the viral DNA get incorporated as spacers in between the short palindromic repeats that we talked about earlier. And the reason it does this is it provides the bacteria with a form of immunological memory. So the immune system essentially remembers what this viral DNA looks like. It's like the CRISPR region is full of mugshots of bad guys. Eventually, those spacers, the mugshots, will get transcribed into a specific type of RNA known as CRISPR RNA. This is kind of like having a photocopy of the mugshot. Now, the CRISPR RNA combines with another type of RNA called tracer RNA. And when they combine, they make something known as the guide complex. This guide complex associates with Cas9 and helps guide the Cas9 protein towards the viral RNA that matches the mugshot. And when Cas9 finds the viral DNA it's looking for, it does its scissors thing and cuts that viral DNA somewhere in the middle. This makes the viral DNA unstable, and eventually it'll just degrade. So, how do we repurpose this to edit genes? Well, some scientists thought to ask, wait, can we hijack this system to cut any DNA we want? And yeah, pretty much. It turns out, instead of relying on the guide complex, that combination of CRISPR RNA and tracer RNA, you just use a synthetic single guide RNA that you designed and has all the stuff you need already fused together. And essentially what you're doing by swapping out the guide complex for your single guide RNA is you're swapping out the mugshots. Don't look for that guy, look for this guy. So now, instead of hunting down viral DNA, the Cas9 protein is going to go after the host's own genome at a site of your choosing. Now, in order to do this, most researchers use some form of transfection to introduce the CRISPR-Cas9 system into the host cells. And transfection basically is a method of manually introducing DNA or RNA into some host cells. So instead of waiting for an infection to happen, you make it happen. For example, you could introduce the messenger RNA that codes for the Cas9 protein along with the single guide RNA that you designed. Okay, so you've got the Cas9 protein, you got the single guide RNA to show where to go, you've cut the host genome. What's next? Well, that really depends on what you want to do. If you just want to silence or knock out a gene, you're pretty much done. Whenever DNA gets damaged, it has a lot of different mechanisms to repair itself. In this case, the DNA will repair itself through a mechanism known as non-homologous end-joining, which is a repair mechanism where DNA is basically like glued back together, kind of haphazardly. In this process, the ends of the DNA get processed by either being trimmed or sometimes filled in, and then they just get stuck back together. And since this process isn't always precise, oftentimes it'll introduce mutations at the cut site, typically making the genes non-functional or knocked out. Why would you want to do this? It's so you can compare gene expression in different cells. Like if you're researching the expression of a specific gene, you're going to want cells that do express that gene and cells that don't express that gene. This way, you can observe any important differences between the cells. A specific example would be if you're researching cancer, you might want to investigate p53, which is a tumor suppressor gene. Now, by observing cells that lack p53 and comparing them to cells that have p53, you can start to understand p53's specific role in suppressing tumors and under what circumstances it carries that function out. Okay, so we've talked about silencing genes. What if you want to add or replace a gene? Then in this case, whenever you're transfecting your cells, in addition to adding the messenger RNA for the Cas9 protein and your single guide RNA, you'll also need to include a repair template, which is kind of like a DNA patch. Specifically, this repair template will need to have the DNA sequence of the gene you want to include. In this case, depending on where the cell is in the cell cycle, it can use a different repair mechanism to repair the DNA, such as homology-directed repair. And in this case, instead of just gluing the DNA back together, it'll actually incorporate that new genetic information that you included in the form of the repair template. Now that gene is part of the genome, it can be expressed normally. And you might take this approach if, say, you've identified a specific point mutation that leads to kidney disease. You can take cells that have that mutation and use this method to replace the mutated genes with perfectly healthy ones. Now, you may have noticed that when it comes to using CRISPR-Cas9 to edit genes, we need the Cas9 protein. We need the single guide RNA to tell the Cas9 protein where to go. And if you're replacing genes, you need that repair template. But you don't need the CRISPR locus. Why is that? Well, we're not archiving anything. We're not using it to build immunological memory. We're not building a collection of mugshots. All we really want to do is make a cut. So we need the instructions, the single guide RNA, and the scissors, the Cas9 protein. Finally, let's address some of the limitations and common concerns surrounding the CRISPR-Cas9 system, at least as far as its use in gene editing. One issue that comes up quite a bit is specificity and off-target effects. In other words, what if Cas9 cuts in the wrong place? Overall, Cas9 is pretty specific and pretty good about cutting where it's supposed to, but it's not perfect. It can tolerate a certain amount of mismatch between the single guide RNA and the genomic DNA where it's cutting. So that is something to consider, because non-specific cutting can lead to real bad things. It can disrupt tumor suppressor genes like p53 or activate oncogenes, which are mutated genes that, when activated, can lead to cancer. And people are working on this concern in a number of ways, such as, first, identifying where these tolerances for mismatch might occur. So at the very least, you might be able to predict some potential off-target cutting. Some people are also mutating Cas9 proteins to make them even more specific, like hyper-specific, essentially making very high-fidelity variants of Cas9. There are also challenges just trying to introduce the CRISPR-Cas9 system into certain cells. Certain stem cells and neurons, which are specialized cells, are highly resistant to transfection, which makes it really difficult to get the system in there in the first place. Also, some bacteria make components that are just too large to easily fit into experimental cells. Another big issue is DNA repair pathway bias. Non-homologous end joining, that one I talked about that it just glues the DNA together, that's a far more active repair mechanism than homology-directed repair. Non-homologous end joining is basically always active throughout all parts of the cell cycle, while HDR is only active during certain stages of the cell cycle. This means trying to add or replace a gene is nowhere near as efficient as just trying to knock one out, because you have to time it to where the cell is in a specific stage of the cell cycle. And finally, there are, of course, the ethical concerns surrounding using CRISPR-Cas9 to edit genes. Is it okay to edit embryonic genes since the embryo literally can't consent? What's to stop people from making designer babies or super soldiers? What's to stop people from using it to make bioweapons? Isn't this playing God? And if so, why are we letting anybody do it? Now, generally, these concerns are being addressed by regulatory bodies like the National Institute of Health or the World Health Organization. But the regulations that they put forth are largely informed by the public. So I highly encourage you, keep educating yourself about CRISPR-Cas9 and other gene editing methods. Find out what are the uses and the potential impacts of using these methods and decide how you feel about all of it. And then let those regulatory bodies know how you feel. Let them know what you are concerned about. And for those of you interested in learning more about the ethical concerns, in the description, I put a link to a Nature article about a case that involved editing germline cells from human embryos that sparked a worldwide moratorium. It's a fascinating read. I highly recommend checking it out. So hopefully I've shown you that CRISPR-Cas9 is amazing. It's a very powerful tool. It can be used to not only study genes, but engineer drought-resistant crops, or even engineer mosquitoes that do everything mosquitoes do, except carry malaria. And who knows, in the future, what kind of diseases that we can treat or maybe even cure using CRISPR-Cas9 or a system like it. So that's it. That's what CRISPR-Cas9 is. That's how we use it to edit genes and some common concerns about it. If you found this video helpful, click like. And if you have any questions or suggestions for future video topics, use the comments. Bye.